when I got invited to speak at this workshop, I got super excited. I was like, but at the same time, I was thinking we only do linear algebra for neural data. So what can we contribute to this conversation? So I decided that I'm talking about that what we've been doing is foundations for foundation models, or discovering the mother manifold. And I will clarify what I mean by that. So essentially, I wanted to clarify what we mean when we and other groups talk about neural manifolds. Of course, everyone has their own definition. But, like at the end of the day, all of us here, I think, want to understand how the brain, the computations that it performs to, mediate behavior and how these are implemented by neural circuits, right? So our idea is, imagine that we have a monkey that, has this magical tree that grows different types of fruits and is trying to decide and plan the movements to grab his favorite food. So if we want to understand that, we think that there's a few things that we can consider and take advantage of to analyze these computations. The first one is that the brain is actually made of neurons, and these neurons have biophysical properties. There's connectivity, neuromodulators, glia, cell types, all these things we've been thinking about, hearing about, neuropeptides, etc. And these form biophysical constraints, which means that your activity cannot be in any state that you want, the activity of your population of neurons, but it will be constrained within a low-dimensional part of this state in which it accesses the activity of a neuron. And this is what we call a neural manifold. And there's a second constraint that matters for this, that is the behavior that the monkey is engaged in. So we combine these neural constraints with the behavioral constraints, and what we have is like this behavioral manifold, that behavior-specific neural manifold that will underpin the computations for this behavior. And our proposal has been, and we've been banging on this drum for almost a decade now, is that neural computation lives at this level of neural populations. Okay. Now this is all philosophizing, but it also, it leads to the following idea, that is that when we are doing an experiment and we record, for example, for 4 neurons or 1,000 neurons, what we are actually, what we can do is take the activity of those neurons and build a state space in which each accesses the activity of one neuron, and we can use dimensionality reduction to estimate basically the neural manifold that we think is an existing entity in the brain. Okay, hope this made sense to everyone. And yeah, this is, this may or may not seem trivial. There was a COSYNE workshop on manifold explained many years ago on whether it was trivial or useful as an idea. But, adopting this idea, our groups and others have done like a few discoveries or contributions. For example, we've shown, and this has come up this morning, that even if the the tuning of single neurons changes a lot across behaviors, even in primary motor cortex, you can identify invariances at the neural manifold level. So we have this paper looking at different hand tasks, and we find that the manifolds from different tasks are actually have similar orientations. They are separated, but they have common components of dynamics, and there's a common readout that explains a lot of behavior. So this already suggests that you can, even with simple linear methods, there's something that suggests there's a foundation model across tasks. Another paper that we put out a few years ago, Matt and me, was, and our colleagues, was showing that actually these manifolds are stable over time. So if we looked at neural activity from monkeys doing the same task over years, we can uncover the same neural manifolds. So these are the activity for different days, and we can just use linear methods to show that this activity is actually at the population level is the same over long periods of time, even if we're recording from different neurons. Okay. And the last thing that we've done in this domain, or one of the last things, is the first topic I want to talk about, that is, what about similarities across individuals, right? So since it's been coffee break, lunch break, and you're probably all falling asleep like me, I wanted to run an experiment, which is ask you to close your eyes and touch the tip of your nose with your right index, please, and leave your hand there. Can you open your eyes and look at your neighbors? So you've all done this, right? I haven't seen anyone miss. But the point I want to make is that this is very trivial, and there's 2 possible explanations for this phenomenon, right? That we can all execute the same behaviors. One is that, we all have like similar bodies. So our body and our spinal cord may impose a bottleneck, and each of our brains may find a different solution to drive our bodies, right, during development. The other option is that we may all have the same solution due to genetically encoded constraints. If that is which seems maybe more sensible. If that is the answer, then the question becomes at what level does this solution live, right? What description level does this solution live? So this is what we looked at in a paper that came out 1.5 years ago, led by Mustafa and John, a postdoc and a PhD student in my lab working with Matt and others. And we started from a very simple idea. We know that the behaviors, since Darwin's grandfather, apparently, I learned this week, that the behavioral repertoire of an animal is defined over evolutionary timescales. And this leads to a genome that ultimately has instructions to build a brain of a certain species, right? So imagine that this is a bucket of monkey brains. I thought it would be weird to put an actual figure here, but imagine these are monkey brains. So each of these brains is unique in their cellular identity, right, at the level of single cells. But what we know is that the way these cells form circuit motifs, columns, layers, regions, so forth, is similar across all monkeys, right? So we know that, and we also speculate based on evidence from a bunch of groups, including Aaron Batista's group here, that there's a relationship between circuit connectivity and these neural manifolds, okay? So maybe we hypothesize what the genome does is specify a manifold where the activity for your different behaviors will live, okay? This is very wishy-washy, but leads to a testable hypothesis, which is that if you have two animals doing the same task, so touching their nose with their index, if it's true that each of these brains instantiate this common neural manifold, then if I record neural activity, and I just do dimensionality reduction to find the neural manifold, I will be able to align this activity across animals, okay? And it also leads to the prediction that as this behavior becomes less similar, these manifolds, these latent trajectories, this activity will become less similar, okay? All clear? So, okay, let's see what we found. The first thing we did was look at simple reaching out tasks that we saw this morning. So we had monkeys doing this task. They were very good at it. You have hand movements here for the two monkeys or two of our monkeys. And here are single neuron recordings. So, they look not very similar by eye, but we don't expect them to because these are different brains, different neurons. But we can do dimensionality reduction and start looking at their features. And you see these are single trials to these 8 targets. And you can see that the trajectories look different, but there's also some common features, right? As they organize, for example, based on target direction, which is color coded. So now the moment of truth is coming. Is like, can I actually take these two sets of trajectories and align them? Okay, as we had predicted, and the answer is yes, as you can imagine. Otherwise, I would be finishing my talk now. And this is even with just like simple linear methods. Of course, it works better if you use like some kind of neural network architecture, but it works very well with linear methods. And we can quantify this, for example, as this correlation as function of the dimensions of the manifold. This goes down because we use CCA, canonical correlation analysis, that basically sorts dimensions by correlation. And these values seem very high. We were very happy, but what do these high values mean, right? We need context. So we did a bunch of lower bound controls that are on the paper. I think the important comparison here is an upper bound, and we compute an upper bound by looking at the same monkey doing this task in the same day, looking at the same neurons, okay? So everything as stable as it could be, and this is what we found, the gradient line. So you can see that these latent dynamics are almost as similar across monkeys as they are within monkey, within day, within neurons, okay? And this is not a cherry-picked example. We found the same for hundreds of comparisons across a bunch of monkeys. Now, this is only looking at these abstract trajectories. Does this tell us anything about behavior? So what we did was essentially do decoding, like what all of us like doing. So here are like the hand trajectories in single trials from one of these monkeys. Just to show you, as we've known from since before I was born, we can decode the position of the hand from primary motor cortex neurons within monkeys. But the cool thing is that we can build a decoder in one monkey, write it in a different monkey, and it works equally well. And again, this is just like linear stuff. And if we don't do the alignment, these dynamics actually look quite dissimilar. Again, this is not just this example, it's all of the examples, okay? So I think this is quite cool because this is unsupervised with behavior, right? We align the dynamics doing the same task and we can decode across animals. So now you can imagine this is why foundation models have to work, if you ask me. Now, the second prediction that I made was that the stability of these latent dynamics should be related to the similarity of the behavior, right? So poking your nose and touching your nose is more different than when two people touch their nose. So to do that, we looked at data collected by our collaborators, Junshol and Josh, from mouse motor cortex as they were doing a reaching and grasping task. And if you look there, you can see that their hand kinematics were less similar across them than in the monkeys. We can show that by computing correlations in hand movements. And you see that the mice movements were much less consistent than the monkeys, which makes sense for a number of reasons. They were trained for less time and mice don't love reaching with one hand. So as predicted, in this case, the similarity of activity across mice was farther away from the upper bound than in the monkeys. But I can show that more directly, basically by plotting the similarity in the neural activity versus the similarity in behavior, and we get this beautiful association between the two. So going back, and by the way, the same is true for decoding. So going back to this idea of foundation models, what this means to me is that we are basically exploring more of this space, right? So we are starting to explore most potential solutions to reaching because we have more behavioral variability. So this moves again toward the direction of multitasking, but not yet. Okay. You may be asking or wondering if these simple linear methods break if you study something that is more complicated than reaching in 8 directions, right? It's a fair concern. So we had this data set of what we call monkey whack-a-mole. So it's essentially monkeys were seeing a sequence of targets. So they will reach one target and all one will pop up and they had to move very quickly, okay, to hit them. And they had to do 4 reaches in a row to get a reward. So the nice thing is that these were all the movements performed by these 2 monkeys in a session. They occupy this bigger space. And then what we did was match these reaches based on the original position and end position, okay? And with that, we could create tens of conditions and do the same analysis, and still we can align fairly well across animals, okay? And there's a bunch of other things in the paper, like comparing microstars, etc. I also wanted to say that this is not only cortex. We found exactly the same in dorsolateral striatum recorded simultaneously in the mice. And just one more thought on this study is that some of you may be thinking, okay, is this trivial because the animals are doing the same movement, they're getting the same sensory feedback? What if the animal is not moving, right? So if the animal is not moving, we actually had that data because we know that animals and humans, before they move, they have to prepare the movement. And this is a fast computation that we know always happens. So we could look at this delay period in which monkeys were waiting for a go cue after they had been presented with the target. So they couldn't move because if they moved, they would get an error and they would have to wait for a penalty period. And we could do the same alignment and it worked equally well. And we could take this aligned dynamics, build a decoder to predict the future reach direction from the monkey from a different monkey. So this is not contingent again on the movement, and this happens with very simple linear methods. Okay, so this tells us that when 2 animals from the same species plan or execute the same movement, they generate the same dynamics, and the similarity of dynamics is related to the similarity in behavior. But if you remember, I started by saying that our intuition for this was that these manifolds were similar because they were built by circuits that had similar connectivity statistics. So what we would like to do, and actually we are doing in a collaboration that Matt Perrick and Adam Hadman are leading, is look at ideally at animals that have a slightly different brains. So for example, neurodevelopmental disorders. But back then we didn't have access to that data. So what we did was build RNNs and manipulate their connectivity to test this, okay? So what we did was train RNNs in the monkey task. We've known how to do this for many years. We basically reproduce the method from David Susilo 2015 paper. And then what we did was add a penalty term to our cost function to make the solution of these networks more dissimilar to that of a reference neural network that we call the standard network. And you can see that as we increase this parameter alpha, our networks did the task, but they develop a bit of tremor. But the interesting thing is that networks with different pairs of networks with the same alpha had a motor output that was as similar as our standard networks. But what we were doing with this is push the networks to explore different solutions in activity space. Okay. So what I mean by that is if you compare align the dynamics of 2 different vanilla RNNs, these dynamics are the same. You can see that they are close to 1. But as you compare the dynamics of pairs of networks with increasing alpha, these dynamics become more and more dissimilar. Okay, so you can have the same motor output with much more dissimilar neural dynamics. Okay, and the way we did that when we looked under the hood, which is the beauty of RNNs, by reverse engineering them and looking at the connectivity statistics, what we see is that basically networks achieve different solutions. In order for networks to achieve different solutions, you need to have different connectivity statistics. Okay. So to recap, I'm going a bit fast, I think. We have found that brains of animals from the same species that perform the same behavior basically do it by generating the same population dynamics. And that behavioral similarity is not sufficient to have the same population dynamics. You also need to have similar connectivity statistics, okay? Now, the piece of the puzzle that is missing here is what happens if you have brains with the same connectivity statistics and solve the task using different solutions, right? But this is hard when you study motor control, like we primarily do, because the solution usually is like, I want to reach here, so I will reach here, right? So my Joanna Lazar, who is a PhD student in the lab now during her master's thesis, which is when she did her project, basically looked for some publicly available data of monkeys doing a decision-making task, okay? This is data from Sébastien Tremblay's Nature Neuro paper. And in this task, basically, monkeys had to learn to associate, they were presented each day with 4 visual cues, and each cue was associated, each target was associated with 2 cues. And they had to learn these associations every day, okay? So the task is very simple. The monkeys would touch the screen. They would be presented with a cue. The cue would go away for between half a second and 1.5 seconds, and then the monkey would see 2 targets, and they would touch the right target to get a reward. Okay? And this was done in freely moving monkeys, which is cool, with nice behavioral tracking, and at the same time, they were recording from prefrontal cortex. Okay? So what we wanted to do first is that if monkeys were doing this task, solving this task in different ways, so what we did was use neural decoding. So for example, this is one of our monkeys in his first sessions. And if we look during the delay period when there's no visual cue, we could decode this cue fairly well during the delay period. And the nice thing is that after a few tens of milliseconds, we can also decode the target very well. So the monkey is prospective in the sense that we can decode what it's going to do in the future. And it's even nicer because this monkey, who was the best monkey at doing the task, there's always one of those. In the later sessions, you could decode the upcoming target from the beginning, right? So he has become better. Then there's another monkey that wasn't that good. And actually, this guy was more retrospective. So you could decode the cue much better than the target in most sessions, but he had a few sessions where he was a bit more prospective. So our prediction would be that the 2 sets of sessions in which the monkeys use the same strategy, which are these 2, we would have more preserved neural dynamics, right? So if you look at that, we can do CCA and we can still align these dynamics across monkeys during the delay period when there's no visual information or anything. You can see that we are still fairly close to the within monkey upper bound and above our control. And the nice thing is that if we compare this to, we add the session in which the monkey was using a different strategy, these dynamics go down. So when monkeys are solving the task in different ways, the dynamics are less preserved, okay? And, we could do this across all sessions. There was a third monkey B, basically do all pairwise comparisons and plot the mean of the first canonical correlations. And we construct a confusion matrix like this. And what we find is some structure that I will just describe quickly based on this that matches the strategies. So we can embed this strategy and the similarity in dynamics matches the strategies. And one last thing about this is that we were interested in knowing one more thing about this task. That is that, we had been discussing a talk by Kia and Obre, who showed that in humans, you can decode from gaze information what they have in working memory, right? So we were like, do monkeys also do that, right? We had all this beautiful behavioral tracking data. So what we did was like try to predict the future target from how monkeys were fidgeting during the working memory period. And we could predict 75% of the time. And these fidgets were actually monkey specific. We can't align within monkeys, across monkeys, but we can align within monkeys. So monkeys fidgeted in the same way every day, but each monkey had their own fidgets. And the nice thing is that, then we asked like, are these fidgets correlated with something that is happening in prefrontal cortex? So when we try to decode the fidgets from prefrontal cortex, we could explain 50% of the variance. And again, this is very monkey specific. Okay. So we were like, okay, we can predict fidgets from activity, fidgets predict upcoming targets. So are decisions embodied, quote unquote? I believe in that we don't do things that are not embodied, by the way. So what we did was define fidget potent and fidget null dimensions in prefrontal cortical activity. And what we found is most of what we could decode about what the monkey is going to do lives in these fidget potent dimensions. Okay. And the nice thing is that if we try to align these fidget null dimensions, there's nothing across monkeys. So it seems that most of what we can pick in terms of what the monkey is going to do and what we can align is related to these embodied fidgets. Okay. And this is it. I've told you about manifolds as a theory of the brain, and we can talk more about it. We have a new manifesto coming up soon. I've told you about this published paper on preserved latent dynamics during motor preparation and motor execution across monkeys, how you actually can break the similarity of dynamics if you manipulate connectivity. And I've told you about this paper that we're hoping to preprint soon, showing that when monkeys solve the same task with different strategies, they basically produce dynamics that are more different. And to our surprise, monkeys may do like some kind of embodied cognition. I wanted to finish, if I have 1 second, with something that I put together during the talks this morning is what does this mean for foundation models? So I think that what our work and that of many other groups has identified these invariances, but also unique features in neural activity, and you can think across tasks, across brain regions, across individuals. And I think this forms the foundation for having a foundation model of the brain. And to me, I think this reflects a mother manifold. And what do I mean a mother manifold? I wrote this during one of the breaks this morning, so I hope it makes sense. It's a mathematical structure that captures the neural constraints imposed by the unique architectures of brains from different species, subject to their behavioral constraints. So this would be, if we were studying all primates, the language of the neural activity of all these primates during all these behaviors. It was basically what Eve and Guillaume and Blake were also talking about. I didn't mention this, but we have papers, we and other groups, comparing this latent neural population dynamics with other modalities like LFPs. And we find commonalities in the activity, but also LFPs have information that spiking doesn't seem to have, which makes sense because they reflect different biophysical processes. So like I think Blake was talking about it, this argues to me that we should incorporate many modalities. And the last thing that I think a few other people also mentioned, like Andreas, I think, and Eve, is that since this, basically these invariances offer a lot of opportunities. One could be defining a computational taxonomy across brain regions within a species, so clearly understand who does what in the brain. Another one could be defining a neurophylogenetic tree. So basically embed animals and all the animals and look at what features are unique and shared. And the last one could be like a functionalist view of development and neurological disorders, where we can identify features of disorders that we could target with fancy tech. And that's it. I want to thank our collaborators, my wonderful lab, and the organizers for having me, and all of you for coming. Thanks.